In this lecture, we are going to be taking a look at the skeletal system, so bones and skeletal tissues, and just open with the definition of osteology, which is the science of bone study. There's a lot of memorization in the skeletal system. You're memorizing the names of the bones and their markings. So I suggest you start looking at your lab information earlier than later, start getting to know all the bones right from left and how you can figure out right from left, as well as the kind of more prominent markings on any of the bones. So we're going to start by talking about bone function. You can see a number of functions listed here. Obviously support, we have our framework for support and our anchoring, right? Like you have pillars for trunk support and just general being able to hold the body upright. It provides protection. So for example, the fused bones of the skull protect the brain, the rib cage protects your lungs. We have body movement. So the skeleton provides anchors for your skeletal muscle, and they act as mechanical levers. So we have a lever system. And then hematopoiesis is dealing with blood cell formation in your red bone marrow. Those are the first four functions. As we continue on with the functions, bones are also for mineral storage. For example, inorganic salts like calcium and phosphorus in the bony matrix. It's going to give weight and strength and firmness to the bones. We also have fat storage in the yellow marrow, which you'll find we talk about in the structure of a long bone in the medullary cavity in the center of the long bone. And then there are some hormones that are produced here, like osteocalcin production, which is going to help regulate insulin secretion, glucose homeostasis, and energy expenditure. So there are about 206 bones in the human body if they're all developed as we tend to see. First, we can classify those by where they're located. And you can see the picture here kind of gives you a general idea of the location. We have the axial skeleton, which is the long axis of the body, including the skull, the vertebrae, the rib cage. It's there for protection, support, and to carry other body parts. And then you have your appendicular skeleton, which is going to deal with your upper and lower limbs and the girdles. And the appendicular skeleton is used primarily for moving and manipulating our environment. So, for example, some things you might see in each, your hyoid, which you find kind of in the neck area, the sacrum, those would be axial skeleton. Things like your calcaneus and metacarpals, metatarsals, all those things you see kind of in blue in the picture, those are the appendicular skeleton. We can also classify the bones by shape. So shape is actually going to kind of determine function, and we have four general shapes. We have long, short, flat, and irregular bones, and we're going to talk in more detail on the next slide about these. On the next slide, you'll get to see a picture of some examples of these four shapes and where you might find them in the human body. But here we're going to discuss how you figure out what bone is what shape. So I already mentioned that we have four shapes. You can see those four names here again, with a basic definition of what they do. So your long bones, like your limbs, those are your levers for movement. They're going to be longer than they are wide. Typically, you have a central shaft with two expanded ends. This would encompass the limbs except for the patella, the wrist, the ankle, and they're mainly compact bone, though we're going to look at the structure of a long bone in a couple slides here, so you'll get a little more information. We have short bones, which are going to be more cube-shaped. They are primarily there to transmit movement forces, so they can alter the direction of pull of a tendon, reduce friction, modify pressure on tendons to reduce abrasion or tearing. So they're pretty important. So things like carpals, tarsals, sesamoid bones, the patella, these are your cube-shaped, and they're mainly spongy bone. We have our flat bones, which are broad, flat, usually curved bones. They serve for muscular attachment or for protection. Things like the scapula, your cranial bones, the sternum, the costals, right, so ribs. These would be your flat bones. They have 2 layers of compact bone with a layer of spongy bone in between them. And then last but not least, we have our irregular bones, which are varying complicated shapes that really don't fit into any other category. They're there to increase muscle attachment, articulation, things like your vertebrae, pelvic bones, facial bones, those would be your irregular bones. So as mentioned before, this slide just gives you a quick peek at some different classifications, locations of different bones. Just a quick look. I would spend a little bit more time getting familiar with how you might classify some different bones, just kind of for your understanding as you move forward with the skeletal system. We have 2 basic types of osseous or bone tissue, and you already kind of identified this on the first lab test. You've seen a picture or a cross-section of bone tissue, but we're going to look at it in more detail here. We have spongy bone, which is deep within the bone itself. It's very porous, red bone marrow, and the trabeculate, which is the framework of bone tissue. And then we have compact bone, which is dense, is going to appear smooth. And here we have, this is your superficial part, but we have organized osteocytes in osteons with an osteonic canal. All right, that was a lot of words. Don't worry, we'll see it again on a later slide. And there's some great pictures in your book. And you can even look it up online and become very familiar with how you identify the structures that we're talking about on this slide. So this is the basic structure of a long bone. Tried to have everything labeled that I could think of, including where you find red versus yellow marrow. So your primary length of a long bone is the diaphysis, kind of that middle portion. When you get to the expanded ends, those are your epiphyses. You're going to have a proximal and a distal epiphysis on any long bone. You can find the periosteum, which is an outer layer. It's an epithelial membrane. The primary kind of superficial portion of the bone is going to be compact, where you can see kind of more of a space-like appearance. That's going to be your spongy bone. On the end, you're going to have articular cartilage. Those are your corresponding ends where it meets with other bones or articulating points. And in the center, the medullary canal is what's going to house your yellow marrow, and we kind of talked about that already. And you can see labeled on here, you have epiphyseal discs or lines, depending on if you are still developing or if that has ended and you are kind of in adulthood. I would spend a little time just making sure that you're aware of the parts and the basic information of how a long bone is structured. And then understand that when you get short, irregular, and flat bones, it actually doesn't have the diaphysis and epiphyses. Those 3 do contain marrow, but they don't necessarily have a marrow cavity, right? So, and again, your red marrow is your hematopoietic tissue, right? We talked about hematopoiesis as a function, so blood cell formation in red marrow. And then our yellow is our fat storage. So understanding where the 2 are located and what they do and how they're different. Your microscopic anatomy is another place I would suggest you spend some time studying, making sure you understand the structure. This can be hard in a recorded lecture. I'll do my best. Your compact bone actually has quite a bit of complex structure, and when we get to look at it a little bit closer, you can actually see that it's set up in repeating osteons, or these sets of rings. And each ring or each osteon is known as your Haversian system, which is the name for both the central canal, the Haversian canal, and the matrix rings. And the central canal is going to run the length of the bony matrix. So you can see in the picture kind of pulled up as one osteon, and those osteons repeat throughout the structure of your compact bone. The very center there is your spongy bone, so don't stress about what's in the very center. We're really looking at this complex compact bone structure. So we have an osteon with a central canal, and the concentric rings that make that up are known as lamella. We also have canals that lead out from the central canal that are the transportation system in the bone, known as canaliculi. And then we also have additionally communication pathways known as perforating or Volkmann's canals. And I would look at some better pictures, but I'll show you another picture on the next slide and we can talk about this again. But you have tiny cavities within this bony matrix, which is the location of your osteocytes, and osteocytes are considered mature bone cells. We're going to talk a little bit more about osteoblasts versus osteoclasts later. This is really kind of a complex structure. So a single osteon has a series of concentric circles known as lamella, and it's around a central canal. That central canal is your Haversian canal. Leading out from the central canal is a transportation system, canaliculi, and we have communication pathways, your perforating or Volkmann's canals. And I know I've repeated that twice. You might have to go back and listen to it again. And I'll point out a few more things on the next slide yet again, because this one is complex and I do want you to spend some time thinking about this and looking at the structure and how to make it work in your head. When I first learned this, it took me a couple tries before I could make sense of the different canals, the central canal versus the canaliculi versus the Volkmann's canal. So making sure that you understand where those are located and what those are for. Okay, here's a bigger look at just the picture from the previous slide. We take a cross section from a long bone and we take a look at it. Now you can more easily see the osteon and the osteonic, in this case, or Haversian canal, the very central. And then it's going to give you yet another look as well. Okay, so from each osteon, if you look at the side cross section there, you can see the perforating or Volkmann's canals for communication. And you can see the osteonic canals with the blood vessels and nerve supply. And then if we take a little tiny closer look, okay, you can see the spaces, the lacuna, and how they house the osteocytes and the canaliculi, right, the communication on that smaller scale. So we've now seen compact bone, spongy bone, right? General anatomy of a long bone, microscopic anatomy of compact bone. And all of these bones of the skeletal system have all sorts of bumps, holes, and ridges, which are all places for attachments. Bones are not smooth, they're very busy, and we have kind of two categories of markings. We have projections or processes, things that grow out from the surface, and typically your projections are attachments of a ligament or tendon. And then we have depressions or cavities, or you could call them openings, which are indentations most of the time for your blood vessels and nerves. A quick general start for how you memorize some of these is that a lot of your projections start with the letter T, which you'll get to see shortly, and a lot of the depressions begin with F. Now that is not all inclusive, but it's a great initial way to start thinking about the difference between a projection and a depression. So we've got projections, things like tuberosities, tubercles, trochanters, and we have depressions, things like fossas and foramen. Now, again, that's just a start. That doesn't, it's not all encompassing, and you're going to see that on the next two slides, but that gives you a start on how to differentiate some of them. So we'll start with the projections, which I kind of already named some of them: tuberosity, trochanter, tubercle. Right, those are listed on here. We also have crest, epicondyle, and process. And they're defined for you here. Some that I want to kind of bring your attention to: this tuberosity. Those are large, rounded projections. Sometimes they're rough. Things like the tibial tuberosity is a good one to pay attention to there. On the crest, you're talking about like the iliac crest. On your trochanters, they're large and blunt, irregularly shaped processes. The greater trochanter you'll find on the femur. And really the only example of a trochanter is on the femur. You won't find it anywhere else. And it is quite obvious when you look at a picture of the femur that you're finding the trochanter. And that trochanter versus the head of the femur is going to be really helpful to you in determining medial from lateral, because obviously you want the head of the femur to go into the hip, so it has to point medially, and then you just have to decide anterior versus posterior. So those are places that'll help you. Tubercles are smaller rounded projections or processes, like the greater tubercle on the humerus. That's not pictured here, but I would pay attention to that. Our epicondyle, we have raised areas on or above a condyle. You have a medial and a lateral epicondyle on the femur. It only shows you the medial on that picture, but you can determine the lateral based on medial because it's going to be the opposite side. And then it kind of gives you like a process as any bony prominence, like a spinous process when you look at the vertebrae. And then the spine, you have a sharp, slender, often pointed projection like the scapular spine. That's a good one to kind of think about there. So those are your projections. And you are going to be naming some of those on the lab test. So I would start kind of learning some of them. Great pictures if you bought the atlas. There's great pictures in there. Otherwise, your book does have some good ones. I'm going to focus on the bottom half of this slide, but the top half has some good examples that are going to help you identify how ribs attach and how your mandible attaches, and what projections help attach that. I would get some familiarity with these, but I'm not going to talk about it here in lecture. So as far as depressions and openings, remember we said this is passageways oftentimes for blood vessels and nerves. We have a meatus on the side of the skull. It's a canal-like passageway. You have sinus cavities within the bone, right? They're filled with air, lined with mucous membrane. You have your fossa. It's going to serve as an articular surface. You can see one of those in the picture. Foramen. Best example of foramen, which you can't necessarily see in this picture, is a foramen magnum. The foramen magnum is a really big deal. It's how your spinal cord passes through connect into the brain. Kind of a big deal there. It does talk about at the top there the projections that form joints. Again, I said I'm not going to spend a lot of time talking about that, but understand that you have to be able to understand the head of the rib and the facets that are there for connecting, and the condyle, which is going to help connect the jaw. I think those are kind of the biggest ones, but again, the foramen magnum is kind of a big deal. You have foramen in the front. It's going to lighten the skull a little bit by making openings and passageways for blood vessels and nerves. So ossification, generally speaking, has to do with bone formation. We have two different types we're going to talk about. We're going to talk about endochondral bone and membranous bone. So the first one we're going to talk about is going to be endochondral ossification. Some basic things to remember is that bone forms from mesenchyme and mesenchymal cells of the embryonic connective tissue. In endochondral ossification, you're talking about bone that develops from cartilage. So we have a number of steps to deal with here, and we're going to talk about all of what I've got listed. So first up, we have our cartilaginous model. So what happens here is we have a cartilaginous model that's initially surrounded by perichondrium. And then ossification is going to actually begin when the underlying mesenchymal cells specialize into osteoblasts. So they're going to start to lay down a bony collar. So it's going to form around the diaphysis of the hyaline cartilage model. This is going to become the primary ossification center, right? So as the bone collar forms, chondrocytes within the shaft are going to enlarge, right? So we get to see right away the primary ossification center. Then we get some decreasing calcification, right? So the cartilage in the center of the diaphysis is going to do some calcifying and develop cavities. And it's the type of deterioration that's going to open up a cavity, but the bone collar stabilizes the hyaline cartilaginous model. So because this is stabilizing and the rest of the cartilage is going to continue to grow briskly, we get the elongation of the cartilaginous model. Then we have our periosteal bud. It's going to invade the internal cavities and spongy bone is going to form. This happens at about month 3-ish. This periosteal bud is a collection of different elements containing a nutrient, artery, and vein, nerve fibers, red marrow elements, osteoclasts. And really the point of the osteoclast here in step 3 is to erode the calcified cartilage matrix. At the same time, we get the activity of osteoblasts that are going to secrete osteoid around the remaining calcified fragments of the hyaline cartilage. So really what's happening here is that bone-covered cartilage trabeculae is our early version of spongy bone and it's forming. And as you look at pictures on the next slide, you'll get to see that. Then with our periosteal bud, we get the enlargement of our primary ossification center. We're going to be spreading hard bone using osteoclasts to break down the spongy bone and form that medullary cavity. Okay, so now we have officially our medullary cavity, and then kind of this is fetal period, week 9 until birth. Our rapidly growing epiphyses consist of only cartilage, and the hyaline cartilage model is going to continue to elongate. And really, ossification is essentially chasing cartilage formation along the length of the shaft. So you get cartilage, and then it's going to chase with your compact bone. Typically, at birth, a lot of your long bones have a bony diaphysis with kind of remnants of spongy bone. But then shortly before, possibly right after birth, we get our secondary ossification center developed in one or both epiphyses. And typically in your large long bones, you get it in both. Smaller long bones, you may only have one. Then eventually, the bones now replaced cartilage everywhere except the epiphyseal plates and the articular surface of the epiphyses. So your epiphyses don't typically ossify until you're about 18 to 20 years old or whenever growth is going to stop. Secondary ossification reproduces really the same steps as primary, except that the spongy bone in the interior is retained and there's no medullary cavity at the epiphyses. And, you know, just you have those epiphyseal plates until they become the line, and that is how, if you break long bones, they kind of take a look at where you are developmentally. We'll look at this one more time on the next slide, so replay this as much as you need, but don't freak out yet. Okay, here we are again. We're going to look at this again. Okay, so you can see your cartilaginous model. Then you get your bone collar forming around the diaphysis of the hyaline cartilage model. Right? Then you can see that the cartilage is going to calcify in the center of the diaphysis and develop into cavities. Okay, so you get your primary ossification center, your periosteal bud, right? So it forms the spongy, then chases the spongy with the compact. You get the elongation going on, secondary ossification sites and longer bones. And then it continues. You can start to see how you have that epiphyseal disc just at the ends. And when it's all said and done, you got yourself a long bone. And in this case, looks like we developed a femur. And you can see just the remnants of the epiphyseal disc or plate that is still cartilage or is now a line and no longer cartilage when it's all said and done. There's a really great picture of this with steps of explanation in chapter 6 in your textbook, figures 6, 10, endochondral ossification and a long bone. It also kind of gives you the timeline. So week 9 is kind of the first 2 pictures. You get to the 3rd month as you get the primary ossification center. And then at birth, you potentially get your secondary. And then it's going to continue from childhood into adolescence. So our intramembranous ossification is a very different process. We're talking about bone that develops from within fibrous connective tissue. Really, we're dealing with flat bone development, things like sesamoids that develop in tendons, so your patella, skull, scapula. So all of the cranial bones are separated by connective tissue membranes during development. Your membranes are called fontanelles. Ossification of these occurs at 20 to 24 months when sutures form between the skull bones. So what's the function of the fontanelles? Well, quite frankly, we need to be thankful because they enable the bony plates of the skull to flex so that the head can pass through the birth canal, and the anterior is closed 9 to 18 months. There's always kind of a soft spot after the birth of a baby, and you have to be careful up until almost 2 years of age with that soft spot and with any of your fontanelles. Again, they fully ossify 20 to 24 months. The anterior one closes sooner at 9 to 18. Sometimes it makes it look like child has a big old forehead. But most bones formed by this process are flat. At about week 8 of development, you get these fibrous connective tissue membranes. And it's kind of 4 steps. You get ossification centers, you get osteoids that are secreted and calcified, you get immature spongy bone and a periosteum, and then you get your compact bone replacing the immature spongy bone. As far as knowing those steps, I don't necessarily require you to know those as much as you need to know endochondral bone development. But if you're interested in seeing kind of more of the details of intramembranous ossification, it is outlined in figure 611 in your textbook or your e-text. So at the epiphyseal plates, we have what are known as growth centers. There's 3 zones. We have our proliferation or growth zone, where you have increased mitosis and chondrocytes. We have a transformation zone. We get larger chondrocytes and columns. This is an area of growth, and we see some changing mineral content. Then we have our osteogenic zone, transforming cartilage into bone. The chondrocytes are going to die. They're broken down by osteoclasts. And then our osteoblasts form osteoid or hard bone. And this is, the osteogenic zone is the location of bone that's going to be adding to the diaphysis. In our diaphysis specifically, we also have appositional growth or bone width increasing. So here we get osteoblasts. Blasts are for building. that are going to secrete bony matrix on the external bone, while osteoclasts, C for consume, are on the internal surface removing bone. So we have both things going on at the same time. Blasts for build, clasts for consume or destroy, if you will. And that's going to change the width of the bone. So bone remodeling is actually continuous throughout life, truly start to finish. You get to recycle 5 to 7% of your bone mass per week. You replace your spongy bone every 3 to 4 years and compact bone approximately every 10 years. Right? So remodeling is really the continuous reabsorption of old bone and the deposit of new bone. And it occurs at the endosteum and the periosteum, so internal and external. And here we are again, our osteoblasts are depositing or building bone, and our osteoclasts are reabsorbing or destroying or consuming bone. We have a thing called Wolff's Law. A bone grows or remodels in response to the demands placed upon it. So bone anatomy is going to reflect the stresses it encounters. And then we have a basic definition of osteoporosis here. I'm going to talk about it again later, but just know that we're talking about osteoclast activity being greater than osteoblast. Make sure you put those in the right order. Hint, hint. Need to know those. Osteoclast greater than osteoblast. So what happens when we break, quote, break a bone? Right? Well, we have steps for healing, and I'm going to go through fracture repair first, and then I'm going to talk to you more about the types of fractures, show you some pictures, show you some examples of some great pictures of some great fractures. So we call it bone fracture repair because it has to do with whether it is complete or incomplete, and you'll get to see that later. But here's the steps of how the repair works. Step 1, we get hematoma formation. So blood vessels in the bone and periosteum are torn. The hemorrhage blood clots forming a hematoma. So the tissue at the site is swollen, painful, and inflamed. Initially, it's not very fun. And I've broken 3 bones in 2020 now, so yay. Different levels of fracture. It's been really fun. I'll tell you more as we get to them. In step 2, we get our fibrocartilage callus formation. Within a few days, the new vessels are going to grow into a clot. Your fibroblasts and chondroblasts are going to invade the fracture site and form kind of the nearby periosteum and endosteum. And they're going to span the break, connecting the ends of where it was broken. So fibroblasts are specifically to produce collagen fibers. Chondroblasts secrete your cartilaginous matrix. So those are the first 2 steps on this slide. We'll get to the next 2. So we have hematoma formation and callus formation. So we're going to have the break, and then we're going to reconnect it and start to protect it. Once we've got our fibrocartilage callus formation, we can move towards a bony callus formation. So osteoblasts begin forming spongy bone. Within a week, the matrix kind of bulging externally. Later, it's going to calcify. So your osteoblasts are going to lay down the trabeculae of the new bone within the fibrocartilaginous callus, spanning the callus, uniting your 2 fragments. And gradually, you'll be replaced by immature bone, leading to the bony callus, and repeating the steps that we saw in endochondral ossification. In the final step, we go through bone remodeling. So during callus formation and for several months after, remodeling is going to occur to lay down and remodel the compact bone so that the new bone looks as much like the original as possible. Just remember that we talked about Wolff's Law, and really it's going to remodel in response to demands placed. And this is why healing fractures follow doctor's orders, because you want it to heal properly. And if you are exposing the fracture to different types of stresses, it may heal differently than intended. So we classify the types of fractures by three pieces of information that we have. So where are the ends after the fracture, the completeness of that break or fracture, and whether or not the bone ends protrude through the skin. So first we have our displaced versus non-displaced. Displaced means that the bone ends are out of normal alignment versus a non-displaced where the bone ends retain normal position. Two of my three recent fractures were non-displaced. One was displaced. So they had to do a little more work on my displaced fracture, but it was on a less, I'd hate to say the word important, but a less impactful bone, so it wasn't quite as big of a deal, though it does ache still. It's been a while. You have your complete versus incomplete. So complete is broken all the way across or through the bone, and incomplete is not broken through. Two of my breaks were complete, one incomplete. Open versus closed. Lucky for me, all of mine were simple, not broken through, closed fractures. Your open ones are the ones that penetrate through. Those are called compound. And then linear versus transverse, those are just exactly what they say. Is it along, which direction is the break? Treatment kind of sucks. All they're going to do is reduce and realign. So they're going to realign the broken bone ends if they need to. If it's open internally, the bone ends are secured surgically with screws and plates. If it's external, the physician's hands are going to kind of coax it back in line. Your immobilization is 6 to 8 weeks for a simple fracture, but because of the time for remodeling, it takes quite a bit longer. I still, two of my fractures still ache a little bit. So I had a complete, displaced, closed fracture of the pinky toe on my right foot. I had a non-displaced, complete, closed fracture of my big toe on my right foot. And I had a non-displaced, incomplete, closed fracture of my left fibula, the smaller bone in the lower leg. So I'm balanced out now. So that's been really interesting. The big toe happened in April while I was teaching from home this Bio 201. And then the pinky toe, 5th digit on my right foot happened July 10, and the fracture of my fibula on the left lower leg occurred on like the 17th of July. I'm pretty good now. I'm going to try to stop breaking things. So over the course of the next, dozen slides or so, the first three here are going to go through the description and the comments and give you a picture of the common types of fractures. And then there's going to be a bunch of examples. Feel free to pause the video and take a look at some of those really good pictures. None of those are of my fractures. I didn't get a chance to pull the discs and load them, so sorry you can't see all my broken parts. But let's talk about these. So we have a comminuted, that's great, comminuted, good luck, you guys just pronounce it for me. Where you get bone fragments into 3 or more pieces. This is super common in the elderly. And you can also see a compression where the bone is crushed. And this is common in vertebrae, especially in osteoporosis. Here we have a spiral, which is a ragged break from a twisting force. This is common in sports. Technically, I had something like that in my pinky toe. Super fun. And then the other one there is an epiphyseal break or an epiphyseal fracture. You get the epiphysis separate from the diaphysis. along the plate itself. And then where the cartilage cells die and calcification occurs. This is the type of fraction that's most significant in growing children, mostly because if you impact the plate, you could slow or stop growth. So that's important to pay attention to. Younger children, this is not a good break because it impacts growth processes. And finally, we have a depression or a depressed fracture where the bone is pressed inward. You see this often with the skull fracture. And then we have a green stick, which is an incomplete break, much like you break kind of a softer twig. One side might break, the other side might bend. This is common in children as well, and kind of because you've got kind of more flexibility in those bones in younger kids than you do in adults. And then now the rest of the slides, as it kind of clicks along, are going to be pictures. Feel free to hit pause and take a look at the pictures. It'll tell you what kind of fracture you're seeing, and it'll give you kind of the x-ray view of those again. Hopefully you found some of those pictures interesting. I didn't say anything during those pictures, so if you were trying to look for talk, there wasn't anything on any of those pictures of different fractures. So we head into bone disorders. I have 4 listed here. The first 2 are both kind of by insufficient calcium or vitamin D. If you increase vitamin D, you could move toward cure, not necessarily get it cured all the way, but osteomalacia is where you get poor mineralization in the bones. The osteoid is produced, but calcium salts are not adequately deposited, so the bones are soft and weak, and this can be quite painful. Rickets is most common in kids. It's more severe than the adult osteomalacia. Here you could see bowed legs, deformities of the pelvis, the skull, the rib cage. Those are pretty common. The epiphyseal plates can't calcify, and then they continue to widen. The ends become visibly enlarged and long. And again, it could be painful. Osteoporosis, we talked about briefly. I gave you the basic definition of osteoporosis already. We're going to revisit that. Osteoporosis is any group of disease in which bone reabsorption outpaces bone deposit, right? So osteoclast activity is greater than osteoblast activity. You get bone mass declining, the bones become porous and light, and therefore they're fragile and they break. 1 in 3 women over 50 have some level of osteoporosis. Some risk factors include decrease in sex hormones, not enough bone stress from just basic daily activity, could be genetics, low calcium, low vitamin D, low protein, smoking, and hormone-related conditions, including some of the things with the sex hormones, but consumption of alcohol or medication at a high level also could lead to osteoporosis. Paget's disease is the excessive bone deposit. It's localized, so It could be haphazard bone deposit and reabsorption. Newly formed bone called poggetic bone is hastily made with a high ratio of spongy bone to compact bone, along with reduced mineralization, causing spotty weakening. Sometimes you get decrease in osteoclast activity and osteoblasts continue. And typically, like I said, it's localized. And last but not least for this lecture, we have some other means of aiding or hastening repair and healing of fractured bones. There are 4 things listed here that may help support bone healing. I've never done any of them for any of my fractures, but that doesn't mean they don't happen. You could use electrical stimulation, skin electrodes on either side of the broken bone, sending in low voltage current to stimulate and accelerate healing. Same kind of idea with the ultrasound, low intensity ultrasound within the first 2 weeks after injury, but before the bony callus forms may help stimulate healing. Vascular fibular graft is harvesting a section of fibular bone with the artery and the vein and transferring it to the femoral head. So that's going to be specific to femur-related injury. And then we have some bone substitutes. And I haven't done too much research on this, but I know they can be super helpful in a lot of places. Calcium sulfate or your gypsum, aka plaster of Paris, calcium phosphate or tricalcium phosphate, which is ceramic based, hydroxyapatite, which is most frequently used, and your bioactive glass, which is bioglass. It's a synthetic silicate based ceramic. Those are all bone substitutes that could be used as a alternative for bone repair. As far as your bones themselves go and the lab itself, I know this is a long lecture. I would make sure you start spending some time memorizing the bones. There are some Kahoots that I will activate as we get along a little further, where you can practice. Please be sure to let me know if it says it's full, because I just have to clone it and reopen it to give you guys as much opportunity to practice. It does have a limited number of people that can play, and then I have to just create another one. And I'm happy to do that to support you. So good luck this week. And I know this was one of our longest lectures. So thank you for kind of tuning in, and then review it, revisit it as much as you would like.